Hello friends, welcome to Science Talk. Today we will talk about arbuscular mycorrhizal fungi or AMF and its role in human nutrition and food safety of humans. Mycorrhiza, as we know, are microorganisms residing in root zone of plants and making symbiotic relationship with almost 80% of plant species. And in this symbiotic relationship, these mycorrhiza, especially arbuscular mycorrhizal fungi, AM or AMF, provides phosphorus nutrition, nitrogen nutrition, and zinc nutrition to these plants, and also they provide sufficient amount of water to these plants, and as a result these plants, the effect of drought stress is reduced in plants which are sufficiently colonized with mycorrhizal spores. Moreover, some studies have also shown that mycorrhizal colonization, when performing efficiently, can also reduce the pathogenic attacks of even foliar pathogens on plants when as compared to those plants which are not efficiently colonized with mycorrhizae. And as a mutualistic benefit, these mycorrhiza get carbohydrates or sugars from plants. Let's discuss what are the factors which can reduce the activity of mycorrhiza in root zone or roots of plant species. One of the main factor is soil with less amount of organic matter. If a soil has less amount of organic matter, it has a negative effect on the mycorrhizal colonization of crop plants grown on such soil. The other factors include phosphorus fertilization. When we provide high amount of phosphorus fertilizers to our crop plants, that will have clear negative effect on the mycorrhizal colonization of these plants roots, because when plants can get sufficient amount of phosphorus without investing their carbohydrate resources in roots, being a very well learned investors, they will not translocate their carbohydrates to roots, and as a result these mycorrhiza will get lazy and they will not make hyphae or mycelium. In fact, the other factor includes flooded rice. When we grow a rice plant in standing water for a longer period of time, it has negative effect on mycorrhizal population in the root zone, because standing water will result in non-availability of oxygen in the root zone, and mycorrhiza, being aerobic organisms, will not be able to thrive under anaerobic conditions, and as a result the plants will not get sufficient or efficient mycorrhizal colonization. That's why it is advised that rice should be provided water in such a way that once we should provide water, then we should dry the soil, then we should provide water again. So in this way there will be more oxygen availability, and as a result these rice plants will get more efficient mycorrhizal colonization. The other factors which can reduce mycorrhizal activity or mycorrhizal efficiency is tillage, because uh, breaks soil, and by breaking soil it also breaks mycelium or hyphae of mycorrhizae, and in order to regenerate these mycelium or hyphae, the mycorrhiza will have to um, invest more energy again and develop this system again, by which can take time, and as a result these plants will have not sufficient for mycorrhizal colonization. One of the other factors which can affect mycorrhizal colonization is crop species in rotation. Sometimes we grow such crop species which are non-mycorrhizal, for example, rapeseed or canola. These species are uh, evolutionary non-mycorrhizal species. They do not make any relationship with mycorrhiza, and when they are grown in a rotation, as a result the mycorrhizal population in the soil also reduced. And as a result, when the next crop is grown, which is mycorrhizal crop, that crop has less mycorrhizal colonization as compared to the similar crop species grown in soil were harvested from mycorrhizal crop species. In this case scenario, I would like to share one of our recent studies in which we show that wheat crop grown after rapeseed has less mycorrhizal colonization as compared to wheat crop grown after wheat. So let's discuss how mycorrhizal colonization can affect human nutrition, because 50% of plants need of zinc is provided by mycorrhiza. So plants grown under such conditions which has less or lower efficiency of mycorrhizal colonization or lower amount of mycorrhizal associations, these crops will have less amount of zinc in their above ground or edible plant parts as compared to those grown under conditions in which mycorrhiza can thrive at efficient rate. So in these crops, these will have more amount of zinc in their edible plant parts. The other effect on human nutrition is actually related to food safety. Mycorrhizal colonization in plant roots reduces the translocation of toxic elements, toxic metals like cadmium from roots into above ground plants. These mycorrhizal colonization or mycorrhizal hyphae can restore cadmium in their cell walls or mycelium and do not allow its translocation into above ground plant parts. And when there is not efficient mycorrhizal colonization, the cadmium available in soil is translocated into above ground plant parts or edible plant parts as compared to those which have higher efficient mycorrhizal colonization. Two of our studies actually clearly showed that the wheat plants which has efficient mycorrhizal colonization has less amount of cadmium as compared to those which do not have ability to colonize mycorrhiza in their roots, because in such soils we have sterilized the soil, and as a result these have limited mycorrhizal colonization, and in the grains and above ground plant parts, these wheat crops has more amount of cadmium in their edible parts. So having crops grown in conditions which can help thriving efficient mycorrhizal colonization will have less amount of cadmium in their edible plant parts as compared to those which are grown in conditions where mycorrhiza cannot thrive efficiently.